think we can start instead of
No,
waiting. So
YouTube
they
का
are
तो
just
चालू हो गया.
joining. We have now
Yes,
72.
it started, sir.
Okay. See how many there in YouTube?
Just a minute. 19 are there on YouTube now.
Ma'am, you can announce just a small, even if you are going to wait, just announce so that YouTube people also come to know that we are going to wait for 2-3 minutes.
Yes, sir. I've already sent the message, sir.
Okay. So ma'am, I'll be now logging it off. If you need something, you can just call me up because YouTube link has started,
Sure,
and
sir.
you can start the recording whenever you are ready.
Yeah. Okay, sir. Okay, there are still some participants who have that gadget names on the screen. I request them to change their name. I think, sir, we can start now. We have 77.
And plus YouTube. Your screen is seen? Hello?
See, sir, we can see the screen. Sir, उसको full screen कीजिए.
My slide you can see?
We can see, sir. वो full screen आप कर दीजिए.
अब तो मैं कर दूँगा बराबर है.
हाँ. Okay, sir. So I am starting to record.
Yeah. Okay.
Okay, so good morning to all those who are present over here. Welcome to the first session of the second day of our workshop. Okay, so without wasting any time, I hand over the session to our moderator, Dr. Lippy Butch, who will be introducing the expert for today. And so that sir can start his presentation very soon. So Lippy, I hand over the session to you.
Good morning, one and all. Good morning, Avia sir. It is my privilege to introduce Professor A.V. Ramchandran sir, ex-Dean, Faculty of Science, MS University, ex-Head, Department of Zoology, Faculty of Science, MS University, Baroda. Professor Ramchandran has 40 years of vast experience in research and teaching. His extensive teaching involvement covers various branches of life science like cell biology, molecular biology, genetics, microbial genetics, developmental biology, reproduction physiology, and endocrinology. Sir has been very actively involved in research in various fields such as developmental biology, metabolic and reproductive endocrinology, toxicology, and so on. He was the first to start the pioneering work on reptilian regeneration in India and initiated studies on female modulation of metabolic functions and reproductive processes in the vertebrate series, with emphasis on birds and mammals. Uh, Sir is also very active in the scientific and academic fields throughout India with various recognitions and awards. The list of recognitions and awards will be a lot to cover in such a short span of time. So, without wasting any more time, sir, I'm sorry I will not be able to do much justice to your profile, but I would request you to. I hand over the session to you, and I request you to start your presentation, sir.
Thank you. Thank you very much, Dr. Lippi. And good morning to all participants. And I hope I'm audible to all of you. Hello? I think those people on the Zoom should be able to answer me.
Yes, sir.
Yeah,
Yes,
because
yes,
I specifically allowed entry into Zoom for my lecture because I find it difficult to talk without interaction, because I think I'm talking to invisible people and I do not know what's going on, whether you understand or not, because I'd like to interact, I'd like to ask questions, etc. That's why. So please do respond, yes or no, whatever it is. Okay? Don't be dummy. There's no meaning otherwise of my allowing some of you to enter through the Zoom. Okay. Now, today's topic, is gene regulation, right? Please be quick. You should keep responding. We're already late. Hello?
Yes, yes, yes.
yes, sir.
So please, somebody or other should keep responding, please. Okay. Now, tell me, do you understand what is meant by gene regulation?
Yes, sir.
Quick.
Yes.
Okay, let me ask you, what is a gene? Come on. You're all master's students trying to appear for the NET examination. The basics should be very clear. Now, tell me, what is a gene? How do you
It's
define a
a
gene?
fundamental unit of DNA.
Fundamental unit of DNA. Okay, what is the fundamental unit of DNA?
Uh,
Is more clearly can you
which
express it?
transcripts and translates into protein, which have some messages.
No, you don't have to go into the details. You can say, when you say fundamental unit, that means it is the structural and functional unit of the genetic material of the DNA, right?
Yes,
The
sir.
DNA is a genetic material. So the structural and functional unit of DNA is a gene. Now, you have another term called cistron. Are you familiar with the term cistron?
Yes, sir.
Okay. Is there any difference between a gene and a cistron, or there any relation between the two?
Cistron is a sequence which will get transcribed and translate, but the main portion.
No. Cistron cannot be transcribed and translated.
Functional unit of DNA.
A gene is also unit of DNA. Cistron is also unit of DNA. Now, I am asking you the difference between cistron and gene, if there is any. What is the exact understanding of a cistron?
A cistron codes for the protein part.
Please do answer loudly.
A cistron codes for the protein part.
Oh, a cistron is a segment of DNA which has coded information within it for a protein. That means it is simply a structural sequence. Now, taking this cistron or the structural sequence alone, you cannot get the protein synthesized, because no transcription and translation be possible. If there is no transcription, there's no question of translation ultimately. So, a gene is a wider term, which is cistron plus something more. Is it clear? A cistron, which has the information for a protein, to get it expressed, you require regulatory sequences. When you talk about transcription, we talk about two important regulatory sequences. One, what you call the promoter sequence, which is ahead of the gene, or you say toward the 5' end. The other is the terminator sequence, which is downstream of the cistron, or you can say to the 3' end. Minus the promoter and terminator, the cistron cannot be transcribed. Is it clear? So a gene is a functional connotation, whereas cistron is only a structure. The structure for the protein is in the cistron, but it cannot be taken out and expressed in the cell unless and until it is flanked by the two regulatory sequences, that promoter and terminator. So a gene is a cistron plus the two regulatory sequences, that is promoter and terminator. Clear? So gene is a functional unit. You can also call it as a transcription unit. Sometimes we use the term transcription unit. That means a unit of DNA or segment of DNA which can be expressed. That means which will be transcribed and translated. In that sense, a gene is nothing else but a transcription unit. Clear? Is it clear now? So that is about a gene. Now, genes, when you say gene expression, it is essentially taking out the information from the gene and getting the end product, mostly protein synthesized for which the gene codes for. Or sometimes the end product could be functional RNA molecules in the form of rRNA or tRNA. That means there are two types of genes. Genes which code for proteins. Most of the genes are protein coding genes, and there are a few genes which code for rRNA and tRNA. Because the rRNA and tRNA are in themselves the functional end products which would play an important role in protein synthesis along with the mRNA. So the product of transcription of protein coding genes are mRNAs, which are intermediary molecules which need to be translated ultimately to get the final functional end product in the form of proteins. Whereas in the case of rRNA genes and tRNA genes, the transcripts themselves are the end product because they are functional molecules. Is it clear? Now the question is, having understood what a gene is, what is gene expression, the question is, what do you mean by gene regulation? Can somebody tell me what is meant by gene regulation?
When the gene should be switched on, when the transcription translation should be switched on, switched off. That's regulating a gene.
Okay, and that means a gene will code for proteins. It can express and get the protein synthesized. But the gene products may not be required all the time. So the gene products should be available in a cell as and when required at the appropriate time, at the appropriate place, in the appropriate concentration. That means though the genes have the information, they can be expressed, but however, they need to be regulated for optimal functioning of the cells, right? So the cells have to decide which genes are to be switched on and when and how long. So there is need for gene regulation. The expression of genes need to be regulated for economic reasons because the cells never indulge in wasteful products. If a particular product is not required, there's no meaning in keeping the gene switched on. So naturally, the gene has to be switched off. Similarly, when required, certain genes need to be switched on. Now, in that context, when we talk about genes and functions, what I call physiological functions, metabolic functions, we can say there are two types of genes. Which are the two types of genes? One set of genes which are concerned with housekeeping functions. Genes mean the genes themselves are not controlling function. Of course, proteins are the workhorses. Obviously, all proteins are coded by genes. And that means when you say the genes controlling housekeeping function, that means the products of those genes, the so-called proteins, mostly enzyme, control the day-to-day metabolic active cell, which is required continuously. So housekeeping functions are those which are required continuously. You may be sleeping, but your cells continue to carry out housekeeping functions, which is required for normal energy generation. Without energy, the cells cannot perform any activities. It cannot be alive. So there are housekeeping genes whose products control housekeeping functions. For example, metabolism of glucose. Glucose is being continuously oxidized to generate. That is a basic source of energy in cells, and we always have glucose in our body. The glucose gets metabolized to produce energy. You know the metabolic pathway involved in that. So this is their housekeeping function, continuous generation of energy to keep your cells alive by performing various activities. That means these are the part of housekeeping gene. So normal metabolic activities which are required to be carried out routinely day in and out, day and night, they are called housekeeping genes. And those genes need to be expressed continuously. That means they're not regulated to a great extent. Maybe in certain quantity terms may be regulated. But there are other sets of genes which are for specialized functions, functions which are not required to be carried out continuously. They need to be perform, these functions are required at certain times in certain places. So naturally, the genes coding for the products which carry out these functions are highly regulated, because some of the products are produced continuously. They may be switched off and not required. There are certain genes which are not being used because their products are not routinely required, but under exceptional conditions, they need to be used, and then the genes are switched on. So there are, when you talk about regulated genes, they are actually for specialized functions of cell. Again, there are two types of regulation possible here. One is one set of genes which are usually not expressed. They are always kept switched off, but occasionally they need to be expressed because there is a need for the function to be performed, in which case occasionally these genes are switched on. Now, such genes are known as inducible genes. Are you familiar with the term inducible genes? Inducible genes are the genes
Yes,
which
sir.
can be switched on when required. Normally, they are in an off state. They are switched off. They remain switched off. Only occasionally they will be required. So only at those times they would be switched on. So that means that the gene is induced. Such genes are induced. The other is what is called repressed genes. See? Now, there are certain genes, for example, amino acids we talk about. Do you think amino acids are required by cells? Do you think amino acids are required by cells? Come on.
Yes, sir.
What
Yes,
more?
sir.
Why? What
Yes,
is the
sir.
yes? What is the need for amino acids?
Sir, we're making the protein, nucleotide.
Protein. There are different types of protein that need to be synthesized in cells continuously. Structural proteins, enzymic proteins. No, the proteins are the workhorses, and all functional cells are controlled by protein. Now, there's a need for continuous production of proteins, and if proteins are to be synthesized, then you require the units of protein called amino acids. So, amino acids need to be synthesized continuously so that protein synthesis can occur. Now, since amino acids are required continuously, now you see the one difference between prokaryotes and eukaryotes. You know what one difference between prokaryotes and eukaryotes, or complex organisms like multicellular organisms, and the simple prokaryotic organism? You know what the fundamental difference? Now, you know, when I take
Membranous structure.
no. I'm coming another thing. You know, I'm asking a question regard to my topic. Now, for example, I take a cell from your body. Okay.
Histone protein.
No. I are
No,
going.
sir. One
tangent
gene,
here.
one cistron relationship.
No.
Sir, in the prokaryotic, in the prokaryotic continuously after transcription, then
No, that is not.
transmission.
No. I'm coming to the fundamental difference. I'm not going into gene regulation at the moment. I'll come to that. I'm asking the fundamental difference in a prokaryotic cell and a eukaryotic. And I'll tell you. I'm giving you an experimental situation. You tell me. I take a cell from your body. Okay. Put it in the culture medium. Given ideal conditions for growth, the cells will multiply, grow, and carry out activities, and give nutrients, etc. Your cells will grow. Yes or no? That's how you have tissue culture. Yes or no? Cells isolated from body can be grown in a culture medium. Given all conditions, necessary conditions, they will grow. Yes or no?
Yes.
Okay.
Yes.
I can take a prokaryotic cell. What is a prokaryotic cell?
Bacteria.
Bacteria.
You are giving an example. What is a prokaryote? The question is, the students are always answer something different from what is expected. You should be answering to the point. When asking you what is your name, you say, I'll stay here, I go this way. No, you only need the name and prokaryote. So what is a prokaryotic cell? It is a primitive cell devoid of a nuclear membrane. Yes, what is it? Somebody telling?
Single cell organism.
All single cell organisms are prokaryotes.
Undefined nucleus.
No.
Undefined nucleus.
So that is why you see, you think before you answer properly. Your concepts have to be clear. All unicellular organisms are not prokaryote. For example, amoeba is a unicellular organism. Is it a prokaryote? No. So prokaryotes are primitive organisms. And what is the primitiveness? Absence of a well-defined nuclear membrane. So the nuclear content, what is the most important nuclear content of a cell? The genetic material in the form of DNA. So this genetic material is not demarcated from the rest of the cytoplasm by the formation of a nuclear membrane. So the absence of nuclear membrane, well-defined nuclear membrane, indicates the primitiveness of cell. So So the prokaryotic cells are unicellular, no doubt, single-celled organisms minus a well-defined nuclear membrane. Now, the rough area in the cytoplasm where the genetic material is present, you call that area as nucleoid. Nucleoid. So nucleoid is the general area in the cytoplasm of prokaryotic cell where the genetic material is present. Okay, that's the primitive genus. Now, I take a prokaryotic cell, say bacterium is an example of prokaryotic cell, put in the culture medium, I can grow them also. Now, do you know the difference? What you need to add in the culture medium to grow a prokaryotic cell and a eukaryotic cell?
Sir, it's basic media.
You call minimal medium.
Yeah.
So in a minimal medium, which cells will grow? Prokaryotic,
Sir,
eukaryotic.
eukaryotic.
Eukaryotic will grow in a minimal medium. Are you sure?
Prokaryotic.
Prokaryotic.
So if I say no, then you automatically the other option is only prokaryotic. You can say that. Right. Only prokaryotic cells can grow in a minimal medium. The eukaryotic cells cannot grow in a minimal medium. We'll come to what the minimal medium, because the eukaryotic are more fastidious. They want all sorts of things ready-made in the medium available, only then they will grow. For example, when you talk about, when you studied biochemistry, you studied about amino acids, you classify amino acids, essential amino acids and non-essential amino acids, yes? Yes or no?
Yes,
What's
sir.
the difference between essential amino acids and non-essential amino acids?
Non-essential amino acids not need to synthesize.
One at a time. What is that?
Non-essential should be provided in the diet.
Non-essential should be provided in the diet.
Vice versa. Essential should be provided.
Essential should be provided in diet.
Essential should be provided.
Essential cannot be synthesized in the body.
Which is non-essential will not be synthesized.
Essential is not synthesized in the body.
Essential is not synthesized. That means they need to be provided
So
in the
they are
medium.
provided in the diet.
So naturally you provide it. So of course, in the organism, it comes to the diet. But if you're talking about the culture medium, when you're growing a eukaryotic cell, you have to provide the amino acid, right? Ready-made. They cannot synthesize. So some of the amino acids only can be synthesized by eukaryotic cell. The others cannot be synthesized. So naturally, you have to provide those amino acids. You need to provide vitamins, for example. You require precursor nucleotides and so on and so forth. So eukaryotic cells cannot grow in a minimal medium, while a prokaryotic cell can grow in a minimal medium. Now, what is the composition of a minimal medium in which you can grow bacteria or prokaryotes? What is the composition, You call it a very minimal medium. A minimal things are added. What are the minimal things? Of course, you require water, right?
And carbon source, nitrogen source, and inorganic salts.
What nitrogen source would you give?
Any nitrate or ammonium salt or something.
No, they are in the form of salts only, inorganic salts only. Nitrogen, everything in the salt only. So
Yeah,
what do you require? A carbon source in the form of glucose, water, plus inorganic substances, inorganic salts, etc. can add. Using these inorganic salts, they will knit together all the essential organic molecules. What are growth promoting? There are 3 essential growth promoting substances that are required for cells to grow. Can you identify the 3 important growth promoting substances? 3. These 3 are essential for cells to grow and survive. Yes?
Carbon, nitrogen, and phosphate source.
You're going very basic into that. I'm not like essential organic elements. 3 essential molecules, growth promoting substances for a cell to grow, whether it's a prokaryotic cell or a eukaryotic cell. Are amino acids growth promoting substances?
Yes.
Yes. What about nucleotides? Are they growth promoting substances?
Yes.
Yes. What about vitamins? Are they growth promoting substances?
Yes.
So these are the 3 fundamental requirements for cells to grow. Nucleotide, all nucleotides available for the cells have to duplicate, multiply, and grow. That means you require a continuous source of nucleotides to synthesize DNA. You require a continuous source of vitamins to carry out various metabolic activities for the cells to survive. You also require amino acids to synthesize protein. These are very important substances. These are called the growth promoting substances. Now, in a minimal medium where you're growing the prokaryotic cells, you're only adding water, glucose, and inorganic salt. You're not adding amino acids, you're not adding nucleotides, you're not adding vitamins, and yet they grow. Whereas you put a eukaryotic cell, the poor fellow does not know what to do. It will not survive. Because it requires, you need to provide amino acids, you provide vitamins, you provide basics of nucleotides, then only they will grow. So that's why eukaryotic cells cannot grow in a minimal medium. It has to be supplemented with essential growth promoting substances, which can avail themselves from the medium and then grow, synthesize proteins, carry out metabolic activities, etc. So the prokaryotic cells, which can grow on a minimal medium, are known as prototrophs. Have you heard the term prototrophs?
Yes.
Yes. So what are prototrophs? These are prokaryotic organisms, the cells which can grow in a minimal medium. By using this minimal medium, they can synthesize all the 3 growth promoting substances. That means all the 4 nucleotides, all the vitamins, and all the 20 amino acids. Whereas the eukaryotic cell cannot do it. Now, unless and until a mutation occurs in a bacterial cell, and the mutation affects one of the genes which is concerned with the synthesis of one or other of the growth promoting substances, in which case the cell might lose the ability to synthesize one of the nucleotides or one of the vitamins or one of the amino acids, in which case what happens is, for such a cell, which is a mutant cell, a mutant for synthesis of growth promoting substance, one or the other, such a cell cannot be grown on a minimal medium, because you must identify the defect in the cell and then provide that substance ready-made. For example, it is a mutation for in the biosynthetic pathway for, say, tryptophan. So it cannot synthesize tryptophan. And actually, all 20 amino acids required for growth and synthesis of protein. So here is a cell which is deficient in tryptophan synthesis. So how do you grow these cells? Now, such a cell is known as an auxotrophic cell. AUXO. Auxotrophic is a mutant cell which is not able to grow on a minimal medium. So in an if you want to grow auxotrophic cells, you must take a minimal medium plus add that substance or those substances, essentially I'm talking about growth promoting substances, which the cell is not able to synthesize. So in this case, in this particular example, the cell is not able to synthesize tryptophan. So you take a minimal medium, add ready-made tryptophan, and put this cell, it will grow. Because what it is not able to synthesize is it is availing from the medium. So auxotrophic strains are mutants which can be grown in what is called a supplemented medium. A supplemented medium is a minimal medium supplemented with the growth substances which the auxotrophic cell is not able to synthesize. It could be a singly mutant or it's a multiply mutant. So depending on all those which you cannot synthesize, you have to provide. That's okay. So that's the fundamental difference. That means using inorganic elements, a prokaryotic cell can synthesize all the amino acids, 20 different. There are biosynthetic pathways for amino acids. Is it clear? So amino acids need to be synthesized continuously. Why went into that ultimately? So I'm talking about one we said inducible system, where the genes are switched on for synthesizing the product. Usually these genes are off. Only when required they're switched on. That's called inducible system. The other is a repressible system, where usually the genes are active, but occasionally it may not be required. So it has to be switched off temporarily. And that means you say the gene is repressed. Repression means inhibiting transcription, preventing transcription, right? So there are repressible regulatory systems and inducible regulatory systems. Now, these are very well studied in the case of prokaryotes, because prokaryotes are simple organisms. It's much easier to study most of the molecular and genetic mechanisms. So we talk about prokaryotes principally. Of course, eukaryotes have cell mechanism is more complicated. We are supposed to cover that also. We'll see whether time permits or not. So when you talk about tryptophan as an amino acid, it has to be synthesized continuously because the cells need to synthesize proteins. So naturally, there must be a ready supply of all the 20 amino acids. So all the 20 biosynthetic pathways should be. Now, remember, I'm talking about prokaryotes now. So all the 20 biosynthetic pathways for the 20 amino acids have to be on in prokaryotic cells. But when there is when the accumulation of an amino, for example, tryptophan in this case, which we'll be studying, there is excessive tryptophan produced, then there is no need to produce any more tryptophan, because the cells will never indulge in wasteful products. They are highly regulated. We could be wasteful, but our cells are not wasteful. We could be idle. Your cells cannot be idle. They're much smarter. They do obey the rules and regulations. So naturally, when there is overproduction of tryptophan, there is abundant availability of tryptophan in the cell. the gene will be switched off. That means it is repressed. So when not required, the gene is suppressed. So the difference between the two is, one is which is off continuously, but when required is switched on. The other is on continuously, but not required, it is switched off. Is it clear? So you have inducible and repressible system. These are two types of gene regulation that you find in prokaryotes. Now, you know the scientists who are studying, and based on who studies the entire process of gene regulation and the concept of operon with reference to prokaryotes came up. It was in 1961. Two workers gave the idea of operon mode of gene regulation in prokaryotes. Can you name the scientists?
Jacob and Monod.
Jacob and Monod. Yeah, the operon concept was put for Jacob and Monod in 1961. They were essentially studying the lack operon in the lactose metabolism. I mean, at the moment, I don't want to use the term operon. They were studying the process of lactose metabolism in E. coli, the K12 strain of E. coli bacterium. It is based on their studies, the concept of operon and operon mode of gene regulation came up in prokaryotes. Now we are introducing the term operon. It is based on the studies of Jacob and Monod, the concept of operons came up. Now, can you define what is an operon? In simple terms, and please remember, your answers should be very simple and don't go into complications. Understand? It is something like, Benz, mujhe maar. Don't do that. So don't go beyond what is asked. So if I ask you to give me a very simple, straightforward, basic answer.
A group of gene.
No. Okay.
Coding for a specific protein.
So see, you will tell all those things. You know, if you ask me what I will say, an operon is a basic unit of DNA, operational unit of DNA. It is a basic operational unit of DNA. That's all I will say. Am I wrong? No, you ask me what is an operon, I will say it is a basic operational unit of DNA. Can you say no? Tell me. Can you say no?
No.
So now you ask further. So what do you mean by the basic unit operation? Then I'll explain. Why should I go into detail which you have not asked me in the first? You asked me what is the operon. I said it is a basic unit of operation of DNA. That's it. Now you let us go into the details. So what is this basic unit of operation? Now, when you say a basic unit of operation of DNA, I already gave you a term transcription unit or a gene. Do you think it is a basic unit of operation? Yes or no? I told you the term transcription unit and gene are synonymous. A gene is a transcription unit, right or not?
Yes.
So it is not a basic unit of operation of DNA? It is. Right?
Yes, sir.
It is, yes.
So the unit of operation in eukaryotes would be single gene. Each gene is operated independently. A gene means consisting of a cistron, a structural sequence coding for a protein, plus a promoter and a terminator. It becomes a transcription unit. This is the simplest unit of operation in eukaryotes. But when you come to prokaryotes, a unit of operation, I'm not saying there are no single gene unit of operation in prokaryotes. There are single gene unit operation. But many, the so-called operons, you see, there is a difference between a transcription unit as a basic unit of operation and an operon unit of operation. Now, you have what are called operons in prokaryotes. What are operons? A unit of a transcription unit which will transcribe simultaneously two or more than two genes, not one gene. Two or more than two genes are operated together. That means you have 3 or 4 cistronic sequences. Each cistronic sequence is coding for a protein. So let us say 3 cistronic sequences. They are put one behind the other. It is contiguous. One behind the other, they are organized together and then regulated by one single pair of regulators. Unlike in the case of eukaryotes, are you able to hear me? Hello? Hello? Are you able to hear me?
Yes,
Yes, sir.
Okay, I thought there was some problem in the net. Okay, fine. What did I tell you now? So in the case of prokaryote, 2 or more than 2 genes can be operated together using one promoter and one terminator. So the unit, this is called an operon. An operon is a unit of operation consisting of two or more than two cistrons regulated by one common promoter and terminator. Unlike in the case of eukaryotes, every gene is operated separately. There is no clubbing together of two or three genes into an operon unit. So you understand the difference between the two now? What is an operon? Now the question is, which genes are clubbed together to operate together in the case of prokaryotes? Essentially, genes whose products are enzymes which are controlling metabolic pathways. They very say, there are single reactions in cell. There are also pathways of reactions. What is the difference between a single reaction and a pathway?
So requirement of more than one enzymes for conversion of reactant to product.
Okay, very simple answer is
pathway is a cascade of reactions.
A single reaction means there is one something which is substrate, something which immediately convert a product. One single transformation produce the end product. Whereas in a pathway, you start with a precursor, then you have a series of reactions which will convert them gradually, step by step, into a final end product which is functional. So a pathway involves a series of chemical reactions which will convert a precursor to a functional end product. In one step, it cannot be produced. It requires a series of steps. Essentially, when you talk about metabolic pathways, they're all series of chemical reactions. Now, there are two types of metabolic pathways.
Yes.
Which are the two metabolic pathways?
Catabolic and anabolic.
Catabolic and anabolic. Now, catabolic pathways are those which are involved in breaking down of the substrate. And anabolic pathways are those which are involved in converting basic precursors into macromolecules, like amino acids, right? So, when you talk about these types of regulation, the two types of regulation, we talk about the inducible and repressible. The inducible pathway, the inducible operons are related with catabolic pathways. For example, you're breaking down glucose, oxidizing glucose. It is a catabolic pathway?
Yes.
Yes. Any sugar you be metabolized, it could be a disaccharide, lactose, arabinose, galactose, anything you break down, that is all catabolism, right? Whereas anabolism is, as you can see in the prokaryotes, using the inorganic elements available in the minimal medium of prokaryotes synthesize the amino acid. So there is what is called amino acid biosynthetic pathway. Since there are 20 amino acids, there are 20 different biosynthetic pathways. So they start with a simple precursor and gradually convert it into the end product in the form of an amino acid. That is an anabolic reaction, an anabolic pathway. Now, usually, when you say two types of regulatory mechanism, prokaryote, inducible systems and repressible systems, the inducible systems are essentially related to catabolic pathways, breakdown of sugars, etc., whereas the anabolic, what is the repressible pathways, are related with anabolic pathways or synthetic pathways, essentially, for example, the synthesis of amino acids. Is it clear? So that is how we study the two things. We'll now go into the details of the inducible pathways by taking an example, that is metabolism of lactose, and we talk about an example of an anabolic pathway which is repressible, that is tryptophan biosynthesis. So here the first slide shows you how genes are expressed and regulated in prokaryotic cells. To understand the regulation of lactose utilization in E. coli, this is an example of a catabolic pathway or inducible pathway. And to understand the regulation of tryptophan biosynthesis, amino acid synthesis, this is essentially an example of a
So we can hear you.
Sir, you are not audible.
I think there's some connectivity issue from sir's side. Let's just wait for a couple of seconds. I think sir is disconnected. We're just checking up with sir. Just please wait. Okay. Sir, can you hear us?
Hello. Yeah, I can hear. You can hear now?
Yes, sir. You are audible.
Share screen now, I think. Okay, you're able to see now?
Yes, sir.
Yes, sir.
Oh my God. Okay. What was it I was telling? I think we finish it. Okay, we go to the next. Now, now look at, we just go through what we already said. Operon, in addition to encoding the information for the specific polypeptide sequence of protein, the genome of the cell codes the information that regulates its own expression. So the operon will not only contain the related cistrons which are organized together along with the promoter and the terminator, but also another gene called a regulator gene, because that is the gene which is going to regulate the switching on or switching off of the operon. Is it clear? So when you talk about lack operon, you have three different enzymes which are coded for by three different structural genes required for lactate metabolism. And these three genes are organized one behind the other, what you call the three structural sequences of citron of lactate metabolism, and they are flanked by the two regulatory sequence proteins and promoter and the terminator. But in addition to that, the operon should also include another gene called a regulator gene, because after all, the operation needs to be regulated. So there is a gene called regulator gene, which is which is responsible for regulating the expression of the operon. Is it clear? In addition to encoding the information for the specific polypeptide sequence of protein, the genome of the cell codes the information that regulates its own expression. So you have what is called a lac regulator gene, which will regulate the expression of lac genes. The three structural genes whose products are enzymes which are involved in metabolism of lactose, they are known as lac genes. So the expression of the lac genes are controlled by a lac regulator. So the genome is divided into sequence of DNA known as genes that have specific functions that we know it. Most of the genes code for synthesis of proteins and some code for RNA that we all talked about generally. We're not bothered about this. Now the genes that code for proteins are known as structural genes, or you call cistrons. Other genes have regulatory functions and act to control the expression of structural genes. So in this case, in the case of lac operon, the lac regulator is the gene which is going to control the expression of the lac operon. Now, the structural and regulatory genes together constitute the genotype and determine the phenotype, that is the actual appearance and activities of the organism. Now, the genes controlling lactose metabolism together with the lac regulator will constitute the genotype. You know what a genotype is? You have studied genetics, right? The genetic constitution for a particular trait is the genotype, right? So these genes are required. The structural gene for lactose metabolism plus the regulatory gene would be required for getting the lactose metabolizing enzymes synthesized in the cell. So the genes are there, that's the genotype, but it's not necessarily the phenotype. Now, what is the phenotype for lac operon? The phenotype is expressed by the ability of the cell to metabolize lactose. When the cell is able to metabolize lactose, the phenotype is excited. Obviously, this is controlled by the genotype. Concerned genes and the regulatory gene together would determine the expression of the enzyme. But if the genes are there, but they're not functional, then the phenotype is not expressed. Is it clear? So the phenotypic expression of the lac operon is the ability of the cells to metabolize lactose, because the lac structural genes are being expressed, the functional proteins are being produced, and they are bringing about metabolism of lactose. The moment you find that the cells are metabolizing lactose, that means the phenotype is expressed. So that is the actual appearance and activities. Now, by controlling those genes of the organism that code for functional enzymes, in the case of lac operon, these three functional enzymes are there, the cell regulates its metabolic activities. They're all generative. Now, some regions of DNA are specifically involved in regulating transcription, and these regulatory genes can control the synthesis of specific enzyme. Now, what is the regulatory sequence we are talking about, which will bring about transcription? What is, you studied, I'm sure, prokaryotic transcription, right? You studied prokaryotic transcription?
Yes, sir.
Yes.
Yes,
So
sir.
what is the regulatory sequence that is required to bring about transcription of a gene? In prokaryotes, of course, or maybe eukaryotes also. What is that? What is a region of DNA ahead of the gene which regulates the transcription? Promoter. So what is the why do you use the term promoter? Why is it so? Why is it called promoter?
Promotes the transcription.
Promoting transcription, because it is promoting transcription. That is why called promoter. So what is the function of the promoter in bringing about transcription? How would it help in bringing about transcription?
Binding of RNA
Cooperate
polymerase.
the transcription.
How would a promoter help in bringing about transcription?
Binding of
It acts as binding site for the enzyme, sir.
What is that?
It acts as a binding site for the enzyme.
Which enzyme?
RNA polymerase.
Binding site for what? RNA polymerase. The promoter is a sequence on which the RNA polymerase can bind. Right? In the case of prokaryote, promoter is the sequence onto which RNA polymerase bind, and then the RNA polymerase moves forward and transcribes gene. Okay. Now, in some cases, gene expression is not subject to specific genetic control. Specific genetic array. What's happening? Shit. What's the problem?
You are audible, sir.
So you are
audible,
audible,
no?
sir.
Okay,
Yes,
you're
sir.
audible. Fine. I
Yes.
Okay. Okay. Where is it gone now? again goes off. Foolish. Join a meeting and sign in. Oh no. What a problem.
You're still
What's
audible,
happening?
sir.
Just a minute. What is happening to me? My site today for the first time is creating problems. Oh, my God. You are audible, but you are not able to see anything. I don't know. All right, come on now.
So we can see your current
Sir,
slide.
we can see your slide.
Your slides can be seen, but I'm
Yes,
not
sir.
able
Yes,
to
sir.
see. No, I
Yes,
got
sir.
disconnected. That's the problem.
I think it's frozen, sir.
Ah. Can you see now? Seen that?
Yes.
Okay.
Yes, sir.
Okay. Now it says in some cases, gene expression is not subject to specific genetic regulatory control, and the enzymes coded for by such regions of the DNA are constitutive. Now, okay. Now let me come to a term constitutive. What do you mean constitutive? Okay, I'll give an example. I told you there are housekeeping functions and housekeeping enzymes, right? Remember in the beginning I talked about talked to you about
Yes, sir.
Yes, sir.
Yes,
now
sir.
I'm saying I'm making
Yes.
please consider my statement. The housekeeping genes are constitutively expressive.
Continuously expressed.
So constitutive means by nature they're always on. It'll be continuously on. So all housekeeping genes are constitutive in nature. That means they are being expressed continuously, not regulated. That means they are continuously synthesized. Oh, it's all there. Now, some enzymes are synthesized only when the cells require them. For example, glucose metabolizing enzymes are constitutive in nature because these enzymes are required continuously to metabolize glucose and generate energy for controlling cell function. Right. Now comes an example where the bacterial cells are forced to use lactose. What is lactose? Because when you're talking about lac operon, it is about lactose metabolism. So we need to know what lactose is. What is lactose?
Disaccharide.
Sugar.
Disaccharide made of what?
Glucose and
Glucose and
galactose.
galactose.
Is a common milk sugar?
Yes, sir.
Yes.
Okay.
Yes.
So now comes a situation where the bacterial cells are getting only lactose. In the environment, they have only lactose. There's no glucose available. What will happen? Will they survive? Okay, I'm giving you two conditions. One, a situation where the cells are having only glucose, they're getting glucose continuously. Another situation where both glucose and lactose are there. And the third situation, only lactose is there. Now tell me what will happen to this. One system where the glucose is available continuously for the cells, bacterial cells. Another system where the cells are having both glucose and lactose available. In the third system, only lactose available. Now what will happen? In the first system,
Second and third.
in the first system,
In the second one, it will take only glucose, not lactose.
So it will not make use of lactose because as long as glucose is available, that is the preferred metabolite. So they will continue to utilize glucose. They will not touch lactose at all. It's not required. Clear? They'll simply ignore lactose. But in the third system, where there is only lactose available, no glucose, now the cells are forced to metabolize lactose to get energy because there is no glucose available. So naturally, they will have to metabolize lactose to survive. So what happened? They will have to switch on the lactose metabolizing genes so that they can metabolize lactose and survive. So only under conditions of non-availability of the normal carbon source in the form of glucose, only then the bacterial cells would use or metabolize lactose. Is it clear? So the lactose metabolizing enzymes, which are a part of the lac operon, will be synthesized only when the cells are forced to metabolize lactose for survival. Only then the lac operon would be switched on. Otherwise, the lac operon remains off, inhibited, repressed. Is it clear? Is it clear?
Yes, sir.
So here is a system. It has to be induced. So when required, what is this condition when required? When it is a question of survival for the cells, no other source available, only lactose available, they are forced to metabolize lactose for survival. Only under this condition, they will switch on the lactose gene. We say the lac operon gets induced. So when the lac operon is called inducible system, we already said. Now when a lac operon is to be induced, now when you say when induced, there should be an inducer. Who will induce it? That means there should be an inducer, right? An inducible system should have an inducer. So there'll be some inducer which will induce, that is switch on the lac operon. So what will be the inducer for the lac operon?
Allolactose.
Allolactose.
Allolactose. What is allolactose? Okay. What is allolactose? What is lactose? You already said it is galactose glucose. Lactose consists of a galactose molecule and a glucose molecule. Hello? Can you hear me?
Yes.
Yes. Because I have problems of connectivity. So when you don't answer, I feel something has gone wrong here. That's the problem. Okay. Tell me, it has got a galactose and glucose. How are they linked?
Yeah.
What is the bond that you find in carbohydrates?
Sir, allolactose is
I'm asking.
having
No. Listen. See, I'm asking a question. You are going to that question. You answer.
glycosidic
What is the type of bond that you?
beta-glycosidic linkage.
Beta-1,6
I
glycosidic
won't say
linkage.
alpha, beta, or nothing. I would say glycosidic bond. I asked you. You asked me a question. What type of bond do you find in carbohydrate? I will say glycosidic bond. That's
Glycosidic
all.
bonds.
I would not characterize further. I now ask you further. Glycosidic bonds could be alpha, beta. Doesn't matter. But glycosidic bonds. So glycosidic bonds are characteristics of carbohydrates, right? So when a glucose and the galactose are to be linked together, you require a glycosidic bond.
Yes, sir.
Okay.
Yes.
What is the glycosidic bond that will be formed between galactose and glucose to form lactose?
Beta-1,4.
Beta-1,4.
Oh, 1,6.
No, beta-1,6.
Beta. Okay. Where do you find beta bonds?
Between
Okay.
two.
Tell me what is the difference between starch and cellulose?
The alpha bond and the beta bond.
Okay. Where do you find alpha bond?
Alpha is in the glycogen.
I don't understand.
Beta will be in cellulose.
Starch is the polymer of glucose.
Yes.
Glycogen is a polymer of glucose.
Yes.
Yeah.
Cellulose is also a polymer of glucose.
Yes.
Yes, sir.
What is the essential difference between these three?
Starch has
Starch contain alpha glucose.
Alpha-1,4.
Which contain alpha glucose? Starch. And
Starch. Cellulose contain beta glucose.
I'm asking you glycogen, you're telling you everything. You considered me, what am I? You only said that. You said starch has got alpha bond. And what about glycogen?
Yes, sir.
Yes, sir. Yes, sir, matlab? Starch contains alpha glycosidic bond. Glycogen would contain what?
Also it contains alpha.
Huh? What?
1,4 and 1,6. Alpha 1.
Arre baba alpha beta baat karte 1,4,1,6 mein khan chale gaye. You mix up things. When you're talking about alpha beta, you talk constantly of that only. You said starch contains alpha glycosidic bond. So I'm asking you, what does the glycogen contain?
Alpha-1,4.
Alpha-1,4.
Alpha glycosidic bond. And starch? And starch? What type of bond will you find in starch? Alpha, beta?
Alpha.
Very good. So, क्या difference हुआ? Starch contains alpha glycosidic bond, glycogen contains alpha, and cellulose also contains alpha glycosidic bond. Is that?
No, sir. Cellulose contain beta.
Okay, beta. Now, when you say glucose and galactose being joined together, is it a starch? Is it a cellulose? Or the part of monosaccharide, disaccharides, and polysaccharides, right? What type of bonds will you find in them? Alpha, beta. Malum nahi? Okay, we'll come to that later as we continue. Just remember that. Now let me continue. Often several enzymes that have related functions are controlled by the same regulatory genes. Example, the lac operon, the three structural genes coding for the lactose metabolizing enzymes are organized together and regulated by common regulatory genes. Now, control of the expression of the structural genes for coordinated metabolic activity in bacterial cells is explained in part by the operon model, which demonstrates how the transcription of mRNA directing the synthesis of these enzymes is regulated. Now, the question is, we said in the case of prokaryotes, number of genes are organized together in one region, one behind the other, and operated as one unit of transcription, right? Which genes will be organized? Are all genes in prokaryotes organized in groups like this, or there are also single gene operation?
Yes,
Already
sir,
talked about. There are single gene
genes.
operations also, but there are also operons. There is no operon mode of organization in the case of eukaryote. The operon mode of organization is characteristic of only prokaryote. Now, when operons are formed, when number of genes are organized together and transcribed as a unit, it is those genes which are having related functions. So, for example, in the lac operon, there are 3 genes. These 3 genes code for 3 enzymes. Now, all these 3 enzymes should be present simultaneously to get lactose metabolized. So, these are called related functions. So, these are the 3 genes having a coordinated function because they have to work together. The 3 products of these 3 genes have to be available simultaneously so that this reaction can go to complete. So, operon is an organization of such related genes controlling a common function or a common pathway. It could be a catabolic pathway or metabolic. They are the ones which are organized together in a unit of operation called operon. Now, this is of great importance for prokaryotes. Why is it that prokaryotes? Now, yeah, for example, same catabolic pathway or anabolic pathway could be operating in eukaryotic cells as well. But in the case of eukaryote, those genes are not organized together and transcribed together. They're all on different chromosomes and transcribed independently. Why so? Why in prokaryote there is such operon mode of organization of related genes and no such operon mode of organization of related genes in eukaryote? What is the explanation?
Because in eukaryotes, transcription is done in nucleus and translation is done in cytoplasm, whereas transcription and translation
So
in
what is wrong
prokaryotes
if
are
single
done
genes are there
simultaneously.
in prokaryotes where acid is being translated, be translated simultaneously? What does it matter? Why they show an operon mode of organization of related genes?
So in eukaryotes, there are both introns and exons.
Now, don't go into that also. Come to
Sir,
a more
maybe
general answer.
due to
That you are
structural
talking about the
complexity.
gene organization, difference in gene organization between prokaryotes and eukaryotes. You say that what you are trying to say is that the eukaryotic cells show a split gene organization where there is no such split gene organization in prokaryote. That's not the answer I'm looking for. Tell
Sir,
me
structural
why there is
complexity.
no yeah.
Structural complexity.
Who structural complex? What? Where?
Sir, as eukaryotes will be having more complex proteins to be expressed and more proteins to produce. So
you are going
it
into
would
more
be difficult.
complex. Eukaryotes are more complex, but you don't make it more complicated. The question was simpler. Again, you're going to more complicated answer you're going to. The simple answer is the eukaryotes are much more DNA required for coding for the protein, so they can have the luxury of operating every gene separately with their own regulatory sequences. Whereas in the prokaryote, the genetic material is limited. It's one single circular DNA molecule. There is a paucity. There is not enough genetic material for the luxury of operating every gene separately. When you operate every gene separately, again, you must have structural regulatory sequences for each gene. Whereas here, when number of genes are organized together using only one promoter terminator, number of genes can be operated simultaneously. So it is a question of economy because the DNA is much smaller in prokaryotic cell, and within that available DNA, they have to organize all their functional genes. So there is no luxury in these cells to waste their sequences, right? Since the eukaryotes are much more DNA than what is required for coding protein, now these sequences can be used as regulatory sequences. So it is a question of the content of DNA. The eukaryotic DNA, we used to, you heard the term junk DNA. Of course, this is a very old term, which you cannot say junk DNA. We said, use the term junk DNA because we never knew anything. So you said,
Yes, sir.
non-functional junk. Now, they're not junk anymore. They're a number of functions, right? So that means, the C-value paradox. You heard about C-value paradox?
Yes, sir.
When you studied molecular value genetics, you heard about the C value paradox. It's really a paradox. No? You heard about
Yes.
C value paradox or not?
Yes, sir.
Yes, sir.
So tell me, can you say what it is?
Different genomes will have different size.
So the general trend is that as the complexity
The
of the
amount of
organism increases, the genome size increases, but this has many exceptions. And because of this, it is known as C-value
Okay.
paradox.
When you start, for example, we talk about the vertebrate series, going towards fishes to mammals, the complexity increases. Say on the evolutionary scale, the complexity increases. As the complexity of the organism increases, you also expect the amount of genetic material also to increase, right? But that is not the case. For example, in the case of amphibians, for example, they got much more DNA what a mammal has. Now you compare the complexity of an amphibian with a mammal, which is more complex? Mammal, right?
Mammals, yes.
So we expect mammals to have much more DNA. But what do you find? Alas, in the case of like frogs and other amphibians, there are much more DNA than a mammalian. That means this is a paradox. So it is not related. The complexity, the amount of genetic material is not related with genetic evolutionary complexity. Is it clear? So that means, in general, even humans, for example, you have much more. Now, you know how many genes are there in human beings? Functional genes? Any idea about the number of functional genes in human beings?
Around 30,000.
Around?
30,000.
30,000. Anything between 30 to 50,000, say about average, nearly about 40,000. Okay. But look at the amount of genetic material is much more than what is required to code for the 40,000 genes. Is it clear? There is a surplus of DNA available. And that is why the eukaryotes have the luxury of operating every gene separately. But in the case where there is a paucity of genetic material within that limited amount of genetic DNA, they have to have all the genes put in. So that is why there is an operon model of organization. So all related genes whose functions required simultaneously, they be operated together. Is it clear? Okay. Now, an operon is a DNA sequence that codes of one or more polypeptides, usually of related function, and a DNA sequence that regulates the expression of these genes. That we already said. Now, here is the example of an inducible system of the lac operon, which is shown here in a diagrammatic representation. Showing both, this is the inducible system. Example could be lac operon. This is the repressible system. It could be a tryptophan operon. What do you find? This is a promoter. This is a promoter for I gene. What is the I gene? I gene stands for the regulatory gene. Right? You say lac I or it. If it is a lactose, you can say lac I gene. So I gene is actually, or you may call it the R gene also. It is coding for a regulatory protein. Is it clear? Now don't concentrate on this word here. I will call this as a regulatory protein. R gene or the I gene codes for a regulatory protein. It has got its own promoter. RNA polymerase binds, transcribes it. You get the regulatory protein. Now, this regulatory gene for a particular operon may not be in the immediate vicinity. They could be far away from the structural sequence of the genes. Is it clear? So, the regulatory gene, for example, lac operon, is far away from the lac operon genes themselves. So, why do I call it a regulatory protein? The I gene or the regulatory gene codes for a regulatory protein. What do you mean by regulatory protein? A regulatory protein is one which will regulate something, right? When you say regulation, it could be positive or negative. There are two ways of regulating anything. A positive regulation or a negative regulation. Is that clear? Are you understanding? Hello?
Yes, sir.
So a regulatory gene or a regulatory protein is a protein which regulates some function. In this particular case, we are talking about the expression of a operon. For example, lac operon. The expression of the lac metabolizing gene will be regulated by the regulatory gene. Now, in this particular case, taking lac operon as an example, what is the role of this regulatory gene? What does it do? This regulatory gene goes and binds to a sequence just ahead of the promoter. You have promoter sequence, and slightly ahead you have another sequence called operator sequence. So operator sequence is like a gate. So the prokaryote binds to that and it has to pass through this gate before it can enter into the structural gene which needs to be transcribed. So the operator sequence is like a gate, and now this regulatory protein of the lac regulator gene goes and binds to the operator site, because this protein has a binding site for the operator sequence of the lac operon. Is it clear? So because of that, this goes and binds there. Once the regulatory gene is bound to the operator, the RNA polymerase bound to the polymerase cannot move forward. The gate is closed, the lac operon cannot be transcribed. This is the normal situation in bacterial cells. A repressor now, because this regulator is repressing the expression of the lac operon gene, you call it as a repressor. This is a lac repressor. So the regulator protein in lac operon is functioning as a repressor, lac repressor, because it goes and binds to the operator sequence, preventing transcription of the structural genes. So this is a repressor. So that is why we say the lac operon is kept repressed. Repressed means switched off, not being expressed. This is what the condition. All the time it remains like this, except a condition where no glucose is available, only lactose is available. The cell is forced to metabolize lactose. Under that condition, the repressor is derepressed. It is removed from the operator. Who will remove or pull away the repressor? In fact, it is not allowing the repressor to bind to the operator. What is that molecule? That is called the inducer. So in switching on the lac operon, when there is extreme necessity to metabolize lactose, an inducer molecule will bind to the regulatory molecule and derepress it. Now, means the repressor is being removed. What is it now? This is the triangular molecule what is shown, which is binding to the repressor protein. This is the inducer. Now, in the case of lactose, you said allolactose is the inducer, right? So the allolactose binds to the repressor molecule or the lac regulatory molecule. That means there are two binding sites on this protein. Remember, we are taking this example as a lac operon. So this is the regulatory gene for lac operon. Since essentially it's repressing the lac operon, you call it the repressor. The lac repressor has to be removed from the operator site or should be prevented from binding to the operator site before the lac operon can be expressed. And that is done by what is called an inducer. In the case of lactose metabolism, allolactose is the inducer. That means this lac regulatory protein has two binding sites. One binding site we already talked about, the binding site with which it binds to the sequence of nucleotides in the operator, and the other is the binding site for allolactose. When allolactose binds to the lac regulatory protein, there's a whenever something binds to a protein, remember, whenever protein-protein interaction occurs, when something binds to a protein, there's always a conformational change. The protein structure changes. Is it clear? So it's actually, when you talk about molecular biology, sequences, conformations, they are the essential regulatory systems of functions. So there's a change in the conformation of the repressor protein when allolactose binds. And because of which the operator binding sequence is masked, the conformation so changes, it is not able to bind to the operator sequence. That means it remains out. What happened? The gate is open now. The RNA polymerase moves forward and starts transcribing the lac gene. Is it clear? Have you understood this part of it? Yes? Hello?
Yes, sir.
Yes,
Now
sir.
look at the repressor system, repression. Again, you have an I gene, regulatory gene, and its own promoter. It again produces a regulatory protein. Now, this is called a repressor protein. But this repressor is usually non-functional. Unlike this repressor of the lac, which is functional because straight away it goes and binds. But this is a tryptophan. This is an example of tryptophan. So this is the tryptophan regulatory gene, which produces the tryptophan regulatory protein, which is essentially a repressor. But this repressor, unlike this, is inactive when synthesized. Whereas this is active as it is synthesized, so it goes and binds to the operator. But this is inactive, so it cannot bind to the operator. Can you see here? The operator is not binding to the operator. So the RNA polymerase bound to the promoter can move ahead and transcribe the gene. Now, this is what is called an inactive repressor. Now, what is required is you require a co-repressor. You require a co-repressor molecule to bind to that. So as against inducer, in inducible systems, in the case of repressible system, you have a co-repressor. The co-repressor binds to the repressor. It now becomes an active repressor, goes and binds to the operator and prevents transcription. Is it clear? So what is now? You require inducer to remove it from the operator site, allow transcription. Here you have to activate the repressor by the binding of a co-repressor so it can go and bind and repress. This is a repressible system, it has repressed. Now, what is this? For example, if this is taken as an example of tryptophan operon, this co-repressor is nothing else but tryptophan itself. The end product of the pathway is tryptophan, biosynthetic pathway of tryptophan. The genes coding for the enzymes of the tryptophan biosynthetic pathway will produce tryptophan as the end product. When the tryptophan concentration builds high, more and more tryptophan is being produced. Now the tryptophan goes and binds to the inactive repressor. That means this inactive tryptophan receptor has a binding site for tryptophan. And when tryptophan binds, there's a conformational change, is now exposes the binding site to the operator side. So this now goes and binds the operator sequence. Is it clear? In this case, when the inducer binds, the operator binding site was masked. Whereas here, when tryptophan binds to that, the operator binding site is now exposed. It goes and binds the operator and stops transcription. Very simple mechanism. This is an inducible system. The top one is an inducible system. Example, lactose. This is a repressible system. Example, tryptophan. Have you understood the difference between the two now?
Yes.
Yes.
Okay.
Yes, sir.
Now,
Yes.
induction and repression are based on regulatory genes producing a regulatory protein that controls transcription by binding to a specific site of DNA. What is that site called? What is the site which the regulatory protein will bind?
Operator.
Operator.
Operator. So operator, because it is why it called operator, because it is that sequence which is going to allow the operation of the operon or prevent the operation of the operon. This basically regulates the operation of the operon. That's why. Now, regulation of transcription may be under negative control. Now, in the lac operon, it is under negative control because the regulatory protein functions as a repressor, goes and binds the operator and keeps the lac operon shut. It is switched off. So that's under negative control. Where mRNA for a particular set of genes are synthesized unless it is turned off by a regulatory protein. Now, this is an example of anabolic or repressible systems, right? Now, the genes are turned off in the anabolic pathways. For example, the synthetic pathway for tryptophan, what is an
Sir, you are not audible. Okay, I think sir has got disconnected again. It does. Let me just connect it back to it. So there's a power cut at Professor A.V. Ramchandran sir's place. So let us just wait for some time if the power can be restored back. Yes, we'll keep you updated. Kindly stay online. Right?
Okay, ma'am. Okay, ma'am.
We'll wait for 5 more minutes for sir to join, because generally power cuts are not resolved very quickly. In the meantime, there is a feedback link in the chat box, and all of you are requested to fill that. Okay, we'll wait for 5 more minutes, and then you'll be updated on the next step. The power cut has not been restored at Professor Ramchand's place. So we shall reschedule the remainder part of his lecture in another slot and shall convey you the details of the rescheduled time and date through email. Please note that we will reassemble for the next session of Dr. Karan Rana at 2 p.m. sharp. Thank you.
Okay, ma'am.
Ma'am, that would be on Zoom or YouTube.
That will be informed to you through the mail. Okay.
Okay. Thank you.
Thank you, ma'am.
Thank you. The next session at 2 p.m. will be on YouTube.